today. And you may recall that the first thing we're going to do is have a Kahoot, because I didn't feel like we had enough time to do it. If you haven't already run into these young ladies, Abby and Britney Hensel, they are conjoined twins who were born, I think in Minnesota, and they are conjoined twins. You can see that they have two different heads, two different brains, two different people, clearly, and their bodies are joined so that one, and I don't remember which is which, one has control of the left arm and leg, and the other has control of the right arm and leg. And they share many body organs. Basically, their spinal columns come down and fuse around the pelvic area. So there's one pelvis, and they are connected. And what I want you to, and you can find out more about them online. They had a reality program for a while, and so they're pretty open about their state. And nobody's going to argue that those aren't two people, right? Clearly they are. What I want you to notice about them, though, and they ride a bike and drive cars and motorcycles and stuff like that. And I want you to think about how they can do that when one consciousness only has control of one arm and the other has control of the other, etc. Interesting question. Anyway, what I want you to notice about them is how beautifully and perfectly melded together they are, right? They are, and there, no cell is out of place, by which I mean, take any one cell in their body, out of their trillions, and all of the neighboring cells are precisely the sort of cell they should be for that kind of a cell. Look in their liver, and every liver cell has got a liver cell next to it. And the melding of their skin, perfectly smooth, no interruption, right? And that is an important clue as to how most metazoans put their bodies together, and we're going to see that. I can't show conjoined twins, though, without talking a little bit about the history of them. You may have heard them referred to as Siamese twins, and there's a reason for that. That's because the most famous conjoined twins were from Siam, what's now known as Thailand. They were Chang and Ing Bunker. It was a last name they took when they came to America. They were born in Thailand, and they got along perfectly well. And as young people, a showman found them and realized that he could pay people money to see these two brothers just interact, right? Basically, a reality show of its day. Only it took place on a stage, and people would pay money to see them and talk to them and stuff like that, right? And so they were pretty badly exploited for several years. Eventually, they found themselves in America, where they were a sensation. Mark Twain wrote a whole comic novel based on those amazing twins, etc. And everyone was totally wowed by them. Eventually, they managed to get away from that manager and have control of their own fate. And so they spent the rest of their lives in the United States, touring the country, and they made quite a bit of money, right? As can happen when you're the one that's in charge of the gate and not some guy who's taken most of it for himself. And they would eventually retire and settle down on a farm in North Carolina, I think, or maybe South Carolina. And they're an interesting story in that, unlike Abby and Britney, notice that they are joined. They each had two arms and two legs. They are joined just in the abdomen. And one myth that people have about conjoined twins is how horrible it would be to be a conjoined twin, to be next to someone who's never apart. And this is you projecting onto them, because, as Alice Drager, in her book, very instructive book, you can tell you, when you find conjoined twins, they're not unhappy about being together at all, right? They are very close, emotionally, psychologically, and they don't want to be separated. And the original conjoined twins make that point because, I forget which, but one eventually died, and a physician, a surgeon, was right there and said, I can cut here and sew you back together and you'll still be alive. And the other brother said, no dice. He wanted nothing to do with that. I don't want to be here if my brother's not going to be here. Right? And as Drager points out, and they would eventually marry they had a big family. They had 21 children, as you can see there. To this date, their descendants still gather, annually. And by now, I have the number there. By now, I think they're in the hundreds. Oh, no, over 1,000 descendants, right? So they did, wait a minute. How did they get married? Well, they bought adjoining farms and they would take turns whose wife they would be with. And when one twin was with his wife, they would put a sheet between them and the other twin would pretend not to be there. And they were perfectly happy with this relationship. And they were very prosperous. They'd made so much money and they were good farmers, turned out too. Question. Yeah, Laura.
One of them died. The other died.
Yeah, I mean, he didn't let the surgeon touch him and he died shortly after, maybe a day or two later, sepsis mostly. Yeah, back there.
But
You'll have to be a little louder.
when they're
Yes, they did. So where are they? Yeah, they did. 4 legs, and so separate pelvises, if that's what you're thinking about procreating. And they had, good.
having
Say it again.
independence of them having healthy.
No. So how do conjoined twins happen? We'll talk about that in a moment. But probably there's no genetic predisposition for that. They were identical twins, so probably they were a monozygotic embryo that split and didn't quite finish splitting. But like Britney and Abby, every cell in their body is next to a neighbor that's appropriate for that kind of cell.
Yeah.
There was another question. No? Yeah, Laura. Nope. And so it's not a mutation. It's not inherited. So these twins tell us about what I call the Metazoan's dilemma. If you're going to be a Metazoan, there's several ways to do it. One is to be a sponge, where in a sponge, it consists of a whole bunch of cells, and they're all pretty much alike. And it grows because one cell divides and sticks nearby, and it keeps growing that way, and that's why sponges are so irregularly shaped. For some forces, right? I mean, there'll be more division going on where there's more matter, more plankton or whatever flowing through the water. So, and that's why sponges only grow in certain places, etc. And it turns out there are a few oddball cells in the sponge, but for the most part, if you're going to make a body where every cell is exactly the same, you got no problem, right? Just keep dividing and don't let go of each other. Right? Mitosis. Simple as that. All the other creatures displayed there, though, have many different kinds of cells. And the fact that there aren't that many sponge species, and there's so many species that have more complicated bodies, tells you which one is more successful, right? There are a lot more species that aren't sponges than there are sponges. And so what happens somewhere in evolution, after our common ancestor was sponges, animals arose that started making 2 different kinds of cells. Some that would end up on the inside of the animal and some on the outside, endoderm and ectoderm, we call it. And some of those animals eventually gave rise to the ctenophores, the C's silent, which are the comb jellies. And here's something, and others of them gave rise to the cnidarians, the jellyfish, hydra, anemones, etc. Right? And there's an interesting thing about all these animals that's different from sponges, which is they all have nervous systems. So it turns out to be a metazoan with a lot of different kinds of cells, you need a good nervous system, right? The sponges and comb jellies have nerve nets, and there's some question about whether, in fact, the nervous system of comb jellies is exactly the same as all the others. But if we leave out the comb jellies, at least, once we get to this point, the cnidarians, everything you know about neurons was already established, right? Action potentials, voltage-gated sodium channels, synapses, chemical synapses, all that stuff, that all got worked out in the common ancestor we share with jelly. And the same genes for making neurons, it's been there a long time. So that also tells you that that's a really successful strategy if you're going to be a metazoan. They all have it, right? And yeah, questions about that. And then things get really interesting once we get away from the jellyfish. There, at some point in the common ancestor we share with all these other animals, some metazoans started producing 3 embryonic kinds of cells. An endoderm that was on the inside, a mesoderm that was in the middle, and an ectoderm that was on the outside. And again, that's a really successful strategy. We'll see later that the first thing an embryo does is to make a primitive gut, basically a hole in the middle of the embryo that's going to be a mouth at one end and an anus at the other, most species. And another landmark happened when the earliest animals are protostomes, which means they make their mouth first. So they make a little dimple in the embryo. We'll see more about it later. And that dimple will eventually give rise to the mouth. We, and lots of other animals you see here, we are deuterostomes. We make our mouth second, which means we make what first? An anus, yeah. Okay, so, we're, insert joke here. Questions about that? We'll talk in more details about that later. So let's talk about, just an example, that the cnidarians, they have behavior, right? Some jellyfish even, right, can avoid light, avoid turbulent areas, stuff like that. And this idea that there are different kinds of cells was one that arose because of the invention of the microscope. Once people invented microscopes and started looking at things really closely, it astonished the world. So Robert Hooke, the British philosopher of the 17th and early 18th century, I think he lived into the 1700s, he got a hold of one of these magnifying glasses, microscopes, and started looking at things like a drop of water from a rain barrel. And the world was astonished that it was full of little creatures. Nobody had ever been able to see them, and so they didn't know they existed, right? And he published a monograph with lots of his drawings. Here's a flea. Everybody knew what fleas looked like, but nobody knew they had little hairs on them. Who would have thought that? Why would a flea need a hair, right? I'm not going to answer that question. And eventually, he took a thin slice of cork and looked at it. And being a plant, of course, it had these cell walls that had survived his treatment. And he called them, they look like cells that monks in a monastery, where each monk has their own tiny little room. It reminded him of that. And so he called them cells. And that's why we call them cells to this day. Questions about that? And, Hooke was a really interesting guy with lots of, he talked about all these animalcules that were in water that nobody knew that were there. So he's really bringing the very beginning of understanding of parasites and things like that. He was also the first to realize, because that guy he knew, Newton, had told everyone about gravity, he was the first one to realize that we are walking about on the face of the earth at the bottom of a vast ocean of air molecules. And it's denser at the bottom because Sir Isaac said, the gravity of the Earth is pulling those air molecules down. Right? So any new invention, and those of you who've been around me before know, I happen to think functional MRI falls into the category. Any new invention is always fascinating, and people start using it, and it's inevitable that people start using the new device and misuse and come to exactly the wrong conclusion. Sooner or later, maybe Hooke, who was the first, people started looking, somebody thought to look at a sperm sample, guess where they got it? And were astonished, again, that there were little tadpoles wiggling around it. I mean, imagine what a mind jolt that was to see that in, right? And then people, as the microscopes got better, eventually some people saw that in the head of the sperm, there was a tiny little body in there. Now the guy who drew this, he believed that he was seeing what he was seeing, right? He was seeing what he wanted to see, because this would explain how development is possible. How can you make a complicated body inside a pregnant woman, right? Oh, it's simple. The complicated body was already there inside the head of the sperm, and it's just, put it in a Xerox machine and keep hitting expand, it'll get bigger and bigger, right? This notion came to be known eventually as Preformationism, right? And another thing, 1694, that supported this idea was when another guy took apart a caterpillar, and he must have been at just the right stage. He found that inside the caterpillar, he could see the butterfly's wings. Oh, that's where the butterfly comes from. The wings are already in there. They've just got to grow a little bit once it makes the transition from pupa to adult, right? And so Preformationism neatly explains how to make a complicated body, right? There's no question left. Oh, it was in there in the first place. But it sort of staggers belief to think, wait a minute. So all of humanity was tucked up inside Adam's left testicle, like nested dolls for infinity, right? And, maybe in a world where you thought the whole world was only 3,000 years old and that people had only been around that long, maybe you could sustain that idea. It would just be another, oh, how wondrous is the creator, right? But eventually, people kept using microscopes and saw that this couldn't quite be true. So they would eventually be able to find embryos. At first, they were sea creatures like echidnas or sea urchins, things like that. They would be able to find aquatic creatures and look at their eggs and see that they were one cell and could actually watch them divide into all those little cells. And so there wasn't a tiny little body tucked up inside that sperm, nor was, by the way, some people looked at eggs and thought they could see tiny little bodies tucked up there too, right? So there was some faction. Well, is everyone's body tucked up inside the sperm or inside the egg? But eventually they could see that everyone starts off with cells and the cells divide. They could see that. And pretty soon you have a sphere of cells, right? And they all look pretty much alike. And this then made people realize that Preformationism had to be wrong, and they had to have some other word to describe it. And the first word they came up with to describe it actually was from Aristotle, who had noticed that he could take chick eggs at various stages after the hen had laid them. And if he opened them up at different times, he could see this unfolding complexity happening in the chick embryo. Right? Clever guy. And he said, he gave a term for this that eventually got translated in English as epigenesis. It is about make. And you can think of epigenesis as a slow unfolding of greater and greater complexity. Got that? And so, we got around the question, we got away from Preformationism, And now we're talking about epigenesis, et cetera, instead. But here's the problem. Nobody had any idea how that happened. Aristotle didn't even try. How is it that first it was just a tiny little speck, and eventually he could see the point where the embryo looks like this. He could see that, and eventually would start growing limbs and stuff. Where did they come from? Right? How is that even possible? And by the way, well into the 20th century, well into today, chick embryos are still a widely used model of development for the obvious reason, right? The hen puts it out, and you can open it up and take a look at it. And these days, you can even dribble a little antibiotic on it, and put tape over the opening, and put it back in the incubator and come back and see it some more later. And I'll give you an insider's tip. The tape needs to be Magic Mend, right? Not just Cellular. 3M Magic Mend. That's what you use. And this increased complexity requires explanation. You can see some more stuff like that online. So what happened, you all know the story of Mendel, right? How he is working with his peas and he discovered genes, et cetera. But maybe you don't know that in his lifetime, he was totally unknown. His work was utterly forgotten well into the 20th century. And then it would be several biologists who would rediscover his findings and realize what it meant. And they ended up causing what's known as the modern synthesis, where they realized that Mendel's important contribution was realizing that traits could be inherited whole as discrete units. And in his lifetime, Darwin's biggest problem for natural selection was one that he never solved, and he could not understand how it was possible. Because people would say, well, wait a minute. Yes, a new trait might arise that would be advantageous, but we all know how breeding is. The offspring, once that individual with the advantage trait mates with someone else, the offspring will have a little bit less of that trait. And after a few generations, it would be totally lost, right? Think of a giraffe with a tall neck, right? It's got an especially tall neck, but it mates with an animal that has a normal sized neck, and the offspring are going to be somewhere in between, the so-called regression to the mean. Everybody knew that's how things work. And Darwin couldn't understand that. But when Mendel discovered that some individuals would get the whole trait and other offspring would not, and some would get something in between, that was when it was possible for that trait to be selected for and to increase in the population. So the modern synthesis is that genes are discrete units of heredity, and that evolution happens when particular genes arise as mutations that are advantageous, and then because the individuals carrying those genes leave more offspring over time in the population, that specific discrete trait comes to increase. How's that? And by the way, nobody knew anything about chromosomes yet, but they were using the term gene already, even though Mendel didn't use it. And yeah, questions about that? Yeah, good. I'm incapable of repeating. I don't remember what I just said. The modern synthesis was that because there are discrete units of heredity, those traits, if they are advantageous, will produce individuals who leave more offspring, and therefore, over time, that trait will become more common in the population. That's nowhere near what I said before, right? But I mean, yeah, and I'm not good at rote, right? So, but if you understand, then you don't have to memorize it, right? You understand? That brings us then to a distinction that'll be important over and over again. They've run into before, I know. And that's the distinction between genotype and phenotype. So your genotype are all the genes mom and dad gave you, and that was set when that particular sperm fused with that particular egg and made a zygote, right? And whatever happened at that monumental event in your eyes, it was a, not cataclysmic, but really important seismic event. At that point, all the genes you're ever going to have were already present, right? And you weren't going to get any more. Now, we can quibble about that some, but in general, the genotype is set at fertilization and never changes throughout your life. Phenotype refers to all the physical characteristic an individual has at one particular point in time. That is its phenotype. When you came out of mom, your phenotype was very different than it is today. And to some extent, while you're sitting here, if you learn anything from me talking to you, your phenotype's changing again, right? Some part of your brain has hopefully remembered something I said. And so your phenotype's not the same, even though your genotype stayed exactly the same. So by the early 20th century, people knew about something called cell differentiation, where they would look at embryos and they would see that at first all the cells looked alike, but as the embryo continued to get bigger and bigger and change, suddenly some cells started to look different from other cells, and they became different. And some are going to make the nervous system, and some are going to make the liver, and some are going to make the lining of the gut, and all that, et cetera. So they could see that the cells were becoming different, and they called that process cellular differentiation, right? Simple enough. But the question is, how do they come to be different? And one theory was that maybe differentiation happens because as cells divide, some cells get rid of genes. I'm going to be a liver cell. I don't need those neuron cells anymore. And so I'm just going to get rid of them, kick them out. And now you end up with a cell that the only genes it has are the genes that are appropriate for being a liver cell. What's it to do? It expresses those genes, makes those proteins, takes on that phenotype, cellular phenotype of being a liver cell, and everything's hunky-dory. And it could have been that way. There's no reason why that's a perfectly viable hypothesis. And it's kind of elegant too, because one thing people notice is that once cells became really different from one another, they never went back. They might they might continue to become more different than everyone else, but they never went back to become, that amorphous glob again. And so, that's one way it could work. And it wouldn't be until the middle of the 20th century that people proved that that can't be the case. And this is an experiment about, and is famous, where people would take an egg from a frog and they would remove the nucleus, basically. And then they would take another cell from a different frog, say a skin cell, and stuck out the nucleus of that skin cell and stick it in this enucleated egg. And what they wondered is, did the skin cell, which was only expressing genes that are appropriate for being a skin cell, did it still have all the genes it needed to make a frog? And lo and behold, at least some of these experiments, right? Lots of the eggs just sit there and die. But at least some of the time, indeed, the eggs started dividing, and they knew that it was using the skin cell genotype because they used an albino donor and put it into the egg of a colored frog, a pigmented centipede. Okay. Everyone get that logic? So the skin cell, it didn't shed any of the genes you need to make toes and frogs, and brains and livers and stuff like that. It had all the genes there. It just wasn't expressing. And so that brings us then to another idea about cellular differentiation. That cellular differentiation takes place as cells in the course of development start expressing a particular set of genes, and therefore that causes them to take on a particular structure, and that causes them to take on a particular function, because they're expressing the genes, making the protein that are appropriate for a liver cell or a neuron or a skin cell. Okay? well, wait a minute. Again, we haven't solved any problem. How do the cells control which genes they're expressing? And that has been the great accomplishment of molecular biology in the last 70 years, right? As a better and better understanding of gene expression. And, and I'm going to go over this quickly because you all know this. What we now know is that you have all 20,000 genes that mom and dad gave you in every cell you've got. And some cells are expressing only some genes and other cells are expressing another set of different patterns of gene expression and therefore different patterns of protein present and therefore different structures, different functions, stuff like that. And the way cells control which genes they're using has to do with the fact that genes are not just a list of amino acids to be put together, but rather that there are promoter and enhancer regions in the DNA, typically said to be upstream and downstream of the structural gene, that are there to listen to the cell's interior through chemical signals to decide whether to turn that gene on or not. Shall we express it or not? So the DNA is a long, complicated bit. And the exons are the parts that actually have information about the amino acid sequence that's going to be used. And that eventually gives rise to nuclear RNA, which can then leave the nucleus after more processing and come up with the messenger RNA that specifies a particular sequence of amino acids and therefore particular structure of the protein that'll get made. And so that's known as transcription. And then the mRNA directs ribosomes and the like to put together a particular string of amino acids in a process known as translation. And another thing we won't really be talking about in this class at all, but is again at the forefront of molecular biology, is after the string of amino acids are made, how they get folded together exactly is crucial for function. And there are all sorts of things also in the cell cytoplasm that are there particularly to cause that newborn protein to get folded the right way so it'll work. All very complicated stuff. And I know almost nothing about it, and I'm not going to talk about it. So, happily you've heard about it some and you're going to hear about it some more. The point is that a given cell, which genes it's going to express depends on what sort of proteins are already inside the cytoplasm. Because there are some things that act as transcription factors, proteins whose whole reason to be isn't to make an axon or a dendrite. The only function that protein serves is to control gene expression. And I wish life were simple and I'd tell you, you know what? There's 25 transcription factors. I mean, if you think about it, as an engineer, a human engineer probably would have only come up with 25 of them or so and found different ways to match me. But in fact, it's bewildering how many different transcription factors there are. We're going to be talking about lots of them. And that's probably a clue that natural selection had to rely on lots of different transcription factors to do something so complicated as cellular differentiation, to actually make an organism that has skin cells and liver cells and neurons, et cetera, right? That took a lot of finagling with gene expression. And so we talk about the upstream region being the promoter region, where one transcription factor might sit there and keep any transcription from happening, but another protein could come along and kick it out of the way, and now suddenly the gene gets expressed. So now we can redefine cellular differentiation yet again. Now cellular differentiation happens when the developmental history of the cell and the proteins that are in its cytoplasm progressively changes which genes are expressed and therefore progressively changes the structure and function of that particular cell. Right? And so that's our best definition of cellular differentiation. Yeah. But there's still a problem, which is, Maria.
So, and in the end, it's like one gene that you express the kind of the kind of things that's like tiny five is what like how I know like those transcription factors have to end up being made like originally like
Yeah. Where does that come? Which came first, the chicken or the egg? What came first, the cell or the transcription factor? Right? Chicken and egg. Well, it was the egg, but transcription factor. Right? And yeah. Let me try helping. And so that means if we follow any particular cell, whatever it's going to be, if we want to understand exactly what it's going to be, all we have to do is keep track of which transcription factors were in it in the first place, right? So go back time one. What transcription factors are present? And I'll remind you that to do the transcription factoring work, the protein eventually does have to go in the nucleus, okay? But so at this time, these transcription factors were present. And given their presence and what they do, that kind of dictates what sort of proteins are going to be expressed at time t plus 1, right? Which is going to influence what happens at time t plus 2. And so every cell in your body has a long, complex developmental history of gene expression. And, it's not magic. What genes it's expressing today has something to do with which transcription factors it made, because they're almost all made within the cell itself, yesterday. Again, I told you, you can't possibly exist. This is too hard. This is too complex. Right? And yeah, good.
so there are different transcriptions.
Many, yes.
How did they get it?
Oh, you can't ask me that question until chapter 2. Okay. Well, okay, I'll give you a hint. Who put those transcription factors in there? How many of you think that was Dad who put them in there when that tiny little sperm had fused with the egg? And yeah, sorry. He contributed DNA and nothing else. When Mom made the egg, she had to make an egg that had the right transcription factors, not only the right transcription factors, but the right transcription factors in the right place within the egg, because the egg might look homogeneous to us, but it ain't.
Like
Good.
project
Yes.
like a completely different set of the outcome is different.
So the important thing in the frog experiment is they took only the nucleus, not the cytoplasm, from the donor and stuck it in an egg that Mom carefully made to have all the transcription factors you need to start making a new individual. That contribution of Mom will lump that together as maternal factors, and they're crucial. You cannot make any creature without having a highly structured egg that has transcription factors, the right ones in the right places and at the right time. And then what's going to happen is a domino effect, because whatever the transcription factors were then, as each division happens, they're going to have an influence on what new transcription factors get made. And there'll be a cascade of transcription factors, and no, it won't be one by one. It'll be fanning out sometimes and coming back together other times, all of which is going to determine what that cell looks like. So now we can understand cellular differentiation. All we have to do is understand gene expression. And by the way, that's at the forefront of molecular biology today and will be for a while to come, I predict. Gene expression is complicated, that control. You know what? I think it's time. I'm putting Don's dentist kids through college. Okay, so now we understand that cellular differentiation, epigenesis, is a result of dividing cells coming to take coming to express different sets of genes, including transcription factors, that will eventually cause them to have different structures, different functions. And now the question is, now we know how to make each individual kind of cell you have, but how do we make sure they're in the right place in your overall body so that they can function well, right? We haven't even addressed that question. And what we're going to find out is there's at least two different ways to do that, to accomplish that. And we'll learn more about that. So, and just in terms of terminology, we'll be talking about cell fate, right? Here's a cell at this stage in development, and it's changing. What is its eventual phenotype? And therefore, what is its fate? And what decides what that particular cell's fate is going to be? So there is a hero in this story, one of my heroes. Oh, yeah. Sidney Brenner, who was from South Africa. Sorry, I meant to. It's not going to let me. Yeah. Yep. One of my heroes, because number one, he was a first generation student like me and many of you in class. And his father was a cobbler. You don't know what that is. That's a guy that makes shoes. And couldn't read. Oh, that's not the one I want. Well, maybe it is. Darn. Okay. Back here. On YouTube, you can find lots of interviews with him. His father couldn't read, but some of the neighbors, there was a nice neighbor lady, had a little more money, and she noticed that Sidney seemed awfully bright. And so she paid to send him to school, right? And he became one of the prominent molecular biologists of the world. You know, won a Nobel Prize in there somewhere, and was an interesting guy in lots of different ways. Anyway, way back in the 1950s, he wanted to tackle the question of cellular differentiation and metazoan development, right? And he realized, already there was a big program of studying fruit flies for heredity, right? So people like T.H. Morgan and those guys who eventually settled in Caltech, they're studying fruit flies like mad, and they're being incredibly successful. They are figuring out things like chromosomes, that the genes are on the chromosomes, that they figure out a way to tell which gene is on which chromosome and which genes are close to each other on the chromosome. And they learn that the genes are arrayed in a straight line on a chromosome. And they figure out how, studying fruit flies of all things, they figure out how to find the distance between genes on a chromosome, so-called morgans, right? Centimorgans, things like that. But the problem with flies for Brenner, and of course, lots of people were studying mice and rats and stuff like that. But the problem with all those organisms for Brenner is that they're too complicated. There are too many cells. How can you keep track of how cellular differentiation takes place across ontogeny, the development of an individual? How can you keep track of that when there are too many of them? And so he said, I want to find myself a nice, tame little metazoan that I can actually understand. And after several years, he hit upon a worm, Caenorhabditis elegans, or C. elegans. It's a flatworm, and everywhere you've been, there's been soil, and in the soil, there's plenty of flatworms of this sort. So they're tiny little microscopic features, and you can barely see them with the naked eye if there's some contrast to them. They live in soil. They are transparent because when you're never exposed to sunlight, what do you need pigment for, right? So they're transparent, and you can put them in a microscope, under a microscope. They get to be about 1 millimeter long. It turns out they can go from a fertilized egg to an individual in about 3 days. So development is fast. You can keep track. And by the way, if you want to go someplace over the weekend, that's okay. Stick them in the fridge. And then when you come back from the weekend, take them out and thaw them out, and now development will continue. So they've got a pause button whenever you want. And they even worked out how to grow them by putting them between two microscope slides that had a thin layer of agar in between it. And you could grow them in there, and the animal would eat the agar, actually. And bacteria, they just suck in the agar, right? And that meant that it was easy to see them. You didn't need an MRI. You just pick up the microscope slide where the animal's living, munching its way through the environment, and stick it on the microscope. Watch it. Right? Don't make the heat. Don't turn the light up too high because you don't want to cook it, but you can keep track of it. And again, if you watched it some today and you want to resume tomorrow, stick it in the fridge, etc. And the other thing that's brilliant about this choice is it turns out that you can see every cell in its body. It needs special microscope attachments, so-called Nemarsky optics, doesn't matter. And you can make out the individual cells. And it turns out they only have about 1,000 cells total. And eventually people would map out their entire nervous system, and it turns out they've only got 302 neurons. And the other thing that's convenient about them is they're hermaphrodites. That is, each individual can reproduce, can self-fertilize and produce clones of itself. So long before anyone knew how to clone anything else, these animals, just keep them by themselves and they'll fertilize themselves and make offspring that are genetically identical to them. Very, right? And what's neatest about them is you can watch the fertilized egg divide and you can keep track of every division. And what astonished everyone, yes. There are a lot of worms in the world. Yeah. So, lots and lots of worms. And they're easy to make fun of. You do know how to tell which end of the worm is rounding. This is why there are so many worm species. They're awfully easy to make. Okay. And maybe you didn't know this. Darwin's last book was another astonishing revelation. As he realized he lived in a world where there's lots of dirt, he was the one that realized all this dirt got made by worms. The only reason we have dirt is because we have worms. Did you know that? Yeah. Okay. And it's a good thing because all the other plants are growing on that stuff. Lots of other plants. Okay. So here is a closer look at the nematode worms, C. elegans. With Nemarsky optics, you can actually make out the individual cells. And so this is what it looks like. There is a front end that with a pharynx, a throat and a mouth and an anus. And they also have eggs inside them. And the eggs actually start dividing before she lays them, before she squeezes them out of her vulva, right? So you can look at a worm and see tiny daughters growing inside it already that will come out. How's that? Questions about any of that? And what astonished everyone as they started watching them is that every worm divided in the same way. There was a first division that was already asymmetrical, and that was a lucky thing because now you could keep track. You could look from one worm to the next and see that first division made a big cell and a little cell. And eventually they came up with terminology for this, where they called the little cell the P cell because they knew as they watched development unfold, that was going to make the posterior end of the animal. And so you can give names to all these cells as they divide, and every worm divides in the same pattern every time until at the end you've got a little bit over 1,000 cells total. And you can watch that unfold. And then here's the other thing that was crazy and wild is you can predict the fate of every cell if you know its mitotic lineage, right? So if you started with P and then became the EMS cell, which became the MS cell and the E cell, the labels are totally arbitrary. And you know that these were going to make the throat, some muscle, and some gonad. And these cells were going to make the intestine. And you only need about 20 cells to make an intestine when you're 1 millimeter long. So what they discovered, and all these cells, so you can make a mitotic division map, mitotic map of every division, and every C. elegans goes through the same step every time. And the fate of each cell can be predicted by its mitotic lineage, which means to say that what genes each cell is going to express can be predicted from the mitotic lineage. And some of the cells are going to be neurons, and they're highlighted in blue here. And what's interesting is, you might have thought that all the neurons would come from the same great-great-grandmother, right? But that's not the case. Some had one great-grandmother, some had another. Sure. There are some similarities, right? Here's a clutch of neurons that all had the same immediate ancestor. But then there are these outliers that happen every once in a while, right? So somewhat different. As people were studying these animals in the 1960s and 70s, they realized that they were watching cell fate, right? That they could predict the fate of every cell by knowing its mitotic lineage. And that suggested a way that cell differentiation could be managed and could be coordinated. And it all comes down to transcription factor. So it turns out the fertilized egg has got this protein, SKN1, that is in one part of the nucleus. And when the division comes, almost all of that protein ends up in P1. And then when that divides, almost all of the protein ends up in EMS. And when that divides again, almost all the protein ends up in the cell that they call MS. And that cell is going to divide and form the throat, the pharynx. So how do you get cellular differentiation? Mom put some transcription factors in the right place. And as division takes place, the transcription factors get divvied up unequally. And these transcription factors are going to cause a cascade of other transcription factors, and therefore a cascade of gene expression, so that you start making the various parts of the various parts you need to make a throat. Everyone can see conceptually how that could work. And they would eventually even make out which genes are being involved, which transcription factors. So, for example, there comes a point where there's some cells that are expressing something called UNC86. I'm not going to talk about the complicated terminology of genes and C. elegans. It doesn't matter, totally arbitrary, but I'm using them in case you run into them. And UNC86 expression causes later the expression of a new transcription factor, MEC3. And then after MEC3 starts getting made, the UNC86 and MEC3 proteins start making dimers, and now they're going to affect expression of yet another set of genes. And eventually, there'll be a whole bunch of genes, we can call them structural genes, that are actually ordinary proteins, not transcription factors, ordinary proteins that cause the cell to take on the right shape and function so that it does its job. How's that? Yeah. So you can see how cell differentiation could work when you only have about 1,000 cells or so. Just carefully put transcription factors in the right places of the egg, which, by the way, also means that there's already predetermined where the divisions are going to take place, right? Where the cell walls are going to form. And then divvy up the transcription factors the right way, have the first set of transcription factors trigger a new set of transcription factors, which then trigger yet another set of transcription factors. And as the cells continue to divide, each individual cell has a rather unique set of instructions, depending on the history of the transcription factors that were in that particular cell before it got here today. Oh, see? Cell differentiation is simple. And not so coincidentally, after all those cell divisions are over, each cell is in the right place it needs to be a throat cell, to form the vulva, right? To form a touch receptor. Did I make that out? What we're seeing, the end result here is a neuron that happens to be a touch receptor, right? So we understand the sequence of transcription factors that cause that cell to become a neuron and to become sensitive to touch. MEC3, that's a protein it uses to detect physical deformation. Now, I'm telling you all this because this whole story has nothing to do with how you got made. In fact, what's happening for C. elegans is kind of the rarity. It's only in really simple, small creatures like this that this system can work. I say that because no one's found a more complicated creature that uses the same system. And you can kind of see how that would be the case. I mean, if you're going to make 1,000 cells, then you can imagine, that you could figure out how to divvy up a few factors. We only talked about one, but there are going to be several. And you could end up with 1,000 different sets of genes being expressed because of 1,000 different pathways of transcription factors, and each one then takes on that function and everything works. But again, like thinking preformationism and that everyone's up in Adam's testicle, when you talk about an organism with trillions of cells, heck, 100 billion neurons alone between your ears, right? You just can't come up with enough transcription factors. And remember, the egg is tiny, right? How much heterogeneity can you put in an egg that small? So that's not how you do it. Question about any of that? Okay. Yeah. I think that's enough today. All right. So when we get back, we'll talk about early experiments that suggested that maybe this kind of cell fate, pardon me, I want to give you one more term. We call this a mosaic determination of cell fate. Mosaic, like tiles in a mosaic that you see. This idea that each individual cell's fate depends on where it came in the mitotic lineage. And as long as the mitotic lineage, as long as the division happens the same way every time, you'll end up with the right cell expressing the right genes in the right place. And what we're going to see is that in more complicated organisms like you, it's not a matter of mosaic control of cell fate. What we're going to find out that in complicated organisms like you, what cell fate they're going to take is totally open for a long time, where almost every cell could eventually become, after the first division that made you, right? Several divisions later, almost any one of those cells could have taken on anything. It was not determined yet. Okay, more later.